Buenas tardes. Bueno, gracias por venir al seminario. Esta vez tenemos un invitado de la Universidad de Oxford. Voy a leer su resumen para que sepan lo que ha hecho y más o menos cuál ha sido su trayectoria en inglés. Y bueno, pues espero pronunciar bien. Our invited speaker today is Professor Renier van der Hoorn. He studied chemistry at Leiden University and focused on plant molecular biology and biochemistry. He completed his PhD in 2001 in molecular phytopathology at Wageningen University in the Netherlands, where he is originally from. He was working on the tomato CF resistance proteins. And then with a Benny grant from the Dutch Research Council, he started his own research by introducing activity-based protein profiling in plants. And he established as a visiting scientist, he established that method in the TSL, in the Sainsbury Laboratory in the UK, which is a very important place for this type of research. He initiated his own laboratory in 2005 at the Max Planck Institute in Cologne, with a consolidator grant from the European Research Council, and moved to Oxford in 2013. as an associate professor and tutor in plant sciences and Somerville College. He became full professor in 2017 with another, he secured another European advanced grant. And his research focuses on improving transient protein expression in plants and using chemical proteomics and structural modeling to uncover extracellular host manipulation mechanisms employed by microbes when colonizing the apoplast. He has many papers. He was telling me only yesterday that last year he submitted 18 papers, which is very impressive. So that's a lot of his. He's a very important person in the field. So welcome, Professor Renier, and thank you. Enjoy the seminar.
Okay, thank you, Claudia. Thanks for introducing me. Also, thanks for picking up a year ago and initiating a collaboration that resulted in the Royal Society fellowship that or funding that allowed me to be here and your student to come to Oxford. So yeah, thanks for being here. I will talk about plant microbe or plant pathogen interactions. And what you see here are 3 leaves infected by 3 different pathogens, all tomato leaves, or tomato leaves. And what they have, these pathogens have in common, they are very unrelated, bacterium, oomycete, and fungi, but what they all have in common is that they colonize the extracellular space of a leaf, which is called the apoplast. And that's a very interesting environment to grow in because there's a change in humidity, temperature, light, but also this is a living surface. The plant responds to infection, and one of the things these plants typically do is to secrete lots of toxic molecules, including hydrolytic enzymes that could cause damage to the pathogen. For many of those cases, we actually don't know what they degrade, but we do know that these pathogens are pathogenic. They have learned to live in this environment, and the way they do that is presumably by secreting inhibitors that particularly inactivate the most harmful hydrolytic enzymes. So this is quite a big and open research field. We do not know what these inhibitors are. We do not know which hydrolases are inhibited. So the mission of the lab is to identify which secreted hydrolases are inhibited. Why is that? And how does it happen? And it will help us to identify novel extracellular components in plant immunity that we can then later engineer to make them less sensitive for these manipulations and so that the plant is less susceptible. Now we focus this research on the model system. This is an interaction between Nicotiana benthamiana and Pseudomonas syringae. This is a strain called DC3000 that was originally isolated from tomato. And the one that we use is the HOP-Q mutant. It's a small detail, but the wild type would trigger an immune response, but the HOP-Q mutant does not. So this is a pathogen that is pathogenic on Nicotiana benthamiana. And a lot of research has been done on DC1000. It's arguably the most investigated plant pathogen in the world. And most of that research is on elucidating how the plant is manipulated by the type 3 effectors that are injected inside the plant cell through a type 3 secretion system. We do not, despite all this investment, not know much about extracellular interactions between these model plants and pathogens. So that is our area of research, and the approach that we take is one hand chemical proteomics, on the other hand off-fold multimer. So we do high throughput in silico predictions of protein-protein interactions to predict interactions between these organisms, and we do reverse genetics to identify the role of these components. And that's really easy, and then you really realize why it's so helpful to work with a model plant and a model pathogen. Okay, so a crucial part in this procedure is to isolate apoplastic fluid because that contains the components that we would like to investigate. The way we do that is to submerge leaves in water, apply vacuum, so you replace the air within the leaf with water, then you take out the leaves, dry the leaf surface with a Whatman paper, and then roll them up like a cigar and transfer into a cylinder. and then you centrifugate at low speed. And at low speed, you spin out the apoplastic fluid without breaking the cells themselves. And you spin them out through a sieve, and there's a collection tube underneath. There are different ways to do that, but this is a larger device that we have. So this is how we isolate apoplastic fluids, and that's a yellow color, a bit of a gold mine for us. This is where it all happens. This is where we are interested in. Now, obviously, in that apoplastic fluid, you basically have two proteomes, one from the plant and one from the pathogen, that interact with each other. So these are antagonistic proteome interactions. But most important is the result of these interactions. So what is the effect of these interactions on the activities of these enzymes? Because the activities of these enzymes dictate the outcome of these interactions. Now, this is a functional proteomic layer of information that we can identify using activity-based protein profiling, which was originally pioneered in medical research by Ben Cravat and Matt Bojo. And it's based on the labeling of proteomes with activity-based probes, which are small molecules that have a reporter. So they are either fluorescent or have a biotin tag, and an inhibitor. And these inhibitors, they mimic the substrate, but they lock the enzymatic mechanism in the covalent intermediate state. So you get a labeling, a covalent labeling of enzymes that were active. So we actually don't look at substrate conversion. That's the classic way of looking at activity. But we look at the availability and reactivity of active sites. And that is a hallmark for proteins being active. The advantages are that we do not need to purify these proteins. We do not need to know the substrate, and we can display multiple protein activities simultaneously in crude extracts and even in living cells. So it's a very powerful technology. Now, just an example, this is a probe that labels glycosidases. This is a probe that we started working with. We got this from the Overclave lab, and this was initially pioneered by Bala Kumaran in my lab, who now is running his own lab in India. So this is a compound that is on one hand fluorescent, so we can detect the protein by fluorescence, or the probe by fluorescence, and here is a reactive group that will react with the active site glutamates of glycosidases. Now, if you incubate this probe with apoplastic fluids isolated from nicotiana and bentamiana and run then the proteins on a gel, you can see that the number of proteins become fluorescently labeled. And by mass spectrometry, we know that this is a bunch of different glycosidases, and here we have a beta-galactosidase that we call bgal1. And you can also see that if you do that with apoplastic fluids isolated from infected plants, we see that the signals are a bit weaker, especially this signal. This signal is much more reduced in infected plants. So we think this might be an inhibitor. Now, to prove that, we do this assay. So we mixed the two labeled samples. And what we see here in this lane is the exact average. That makes sense because you mix the two labeled samples. Yeah? We can quantify that. And indeed, that signal that we get in that third lane is exactly the mean of the two first samples. However, if you now first mix the samples, the apoplastic fluids, and then do the labeling, then you get less signal. So that means there is something in the apoplast of infected plants that is able to suppress the activity of the labeling of the enzymes that are in the non-infected plants. So this trick is called convolution ABPP, and it is for us an important clue for looking for an inhibitor. So that raises two main questions, the two that I mentioned earlier. Why is this inhibited and how is it inhibited? And I'm going to address both of those questions today. So first, why is it inhibited? Does it act in immunities? Is bgal inhibited because it acts in immunity? So to test that, we made knockout lines that lack this enzyme with genome editing. And when we isolated apoplastic fluids from these plants and looked at beta-galactosidase activity, we noticed there's a massive reduction in that activity in the apoplast. So it is the major beta-galactosidase activity. And we did that with a fluorescent substrate that becomes fluorescent when you remove the galactoses at these two ends. Okay, so we have a knockout. There's no growth phenotype, but when we put the pathogen on these plants, there is a subtle but significant susceptibility phenotype. So these mutants are slightly more susceptible for bacteria. And actually the bacteria, of course, that we use are the DC3000 strain. So that makes sense. DC1000 suppresses an enzyme that is actually involved in immunity, right? So how does it work then? What does this enzyme do to cause that immunity? So initially we thought, okay, this is a hydrolytic enzyme. It may directly affect the biology of the pathogen. Maybe it breaks down the cell wall or other components. But we did never see, in several assays that we did, we could not detect an effect on bacterial growth. So we then hypothesized that maybe this is a phenotype that occurs only during infection. For example, a B-gall might release something from the bacteria that is recognized by the plants. And that would then induce an immune response that would explain why B-gall acts in immunity. Now, typically, if you do this type of assays, if you want to detect something that is released from a pathogen, you would monitor the release of reactive oxygen species from the plant. So that is the assay that we then established. So what we did here is we mixed Pseudomonas syringae with apoplastic fluids containing B-gall or not, and then added that to a leaf disc of a plant floating on luminol HRP. If the leaf disc recognizes an elicitor, then we get the release of reactive oxygen species that are detected with chemiluminescence in the well and that we can detect in a plate reader. Now, and indeed, something is released from these bacteria because we suddenly get a very nice transient release of reactive oxygen species. This is a typical oxidative burst that a plant will do when they recognize something. And this is dependent on B-gall because if in the absence of B-gall, there is no release of such an elicitor. Okay, so that is done when you take leaf discs of Nicotiana benthamiana. But if you take leaf discs of Arabidopsis thaliana, you also get this recognition event. That indicates that something is released that is recognized by both plant species. So it's probably a conserved elicitor. Of course, we thought, okay, at that stage, this is a beta-galactosidase. It probably acts on a glycan. So we need to look at glycan receptors. So we knocked out we studied lots of mutants of Arabidopsis that lack different glycan receptors and found no effect. But we included at some point also FLS2. And FLS2 mutant of Arabidopsis is blind for this elicitor. So it cannot sense this elicitor. Now, FLS2, as you may all know, is a well-studied receptor kinase that senses flagellin. So that indicates that the elicitor that is released from the bacteria is actually flagellin. And we could quickly demonstrate that by using a flagellin mutant that cannot make flagellin. And indeed, these bacteria do not release the elicitor in the presence of B-gall. Okay, so we know what the elicitor is, but that still raises a question: how on earth does a glycosidase release a peptide-based elicitor? Now, that is because flagellin is hidden. The part that is recognized is a 22 amino acid peptide called FLG22, which is deeply buried into the polymer of flagella. And these flagella are covered with a glycan because every monomer has 6 glycans. Yeah? And those glycans are consisting of 2 or 3 rhamnoses and a modified viosamin. We know this because Yuki Ikenose spent his whole career on identifying and characterizing these glycans. So he showed that for DC1000, but admittedly he actually did most of his research with a different strain, the 6605. The reason for that is simply because this is a bacteria that swims better, it produces more flagellin protein, so if you do research on flagellin, it will go better with this strain. Now, good thing is that this strain is a pathovar tabaci, so it actually also infects Nicotiana benthamiana, and interestingly, it actually does not make that bGAL1 inhibitor. So phenotypes that we were seeing are probably more visible in these mutants. So we started to collaborate with Yuki to get access to all these mutants. And at first we needed to test if this pathovar is behaving like the DC1000 that we studied before. The wild type has flagellin, that is glycosylated in the same way as DC1000 has it. Yeah? And indeed, if you incubate these bacteria with apoplastic fluids containing B-gall, then we do release an elicitor, and that's much less in the absence of B-gall. And these mutants, B-gall mutants, are more susceptible for this strain. So B-gall acts in immunity also against this strain. So then we took advantage of a mutant that cannot produce the glycan. So it still makes flagellin, but it is not glycosylated. This is a flagellin done because we have a mutation in the flagellin glucosyl transferase gene. Now, if you incubate this mutant bacteria with apoplastic fluids, and then add it to a leaf disc, you see immediately a very strong oxidative burst, and that is independent of the presence of B-gall. That makes sense because it's basically the protein is no longer covered with the glycan, so it's instantly recognized. And consequently, these bacteria don't grow very well compared to on wild-type plants, yeah, because they instantly trigger an immune response. But more importantly is the fact that in that background, on these mutants, B-gall mutants do not have this additional, do not support increased bacterial growth. So that indicates that B-gall acts in immunity only when there is a glycan on the flagellin. So then we also took advantage of a couple of mutants. One of them is VOT, which does not have that, it still has the rhamnosis on the flagellin, but does not have that modified viosamin. And very similar to the FTT1 mutants, these mutants are instantly recognized, and they don't grow very well, and the B-gall mutants are not more susceptible for this mutant. So that indicates that B-gall only acts in immunity when the bacteria carry a modified viosamin on the flagellin glycan. And that makes a lot of sense because galactose has structural similarities with this modified viosamin. Now, as another kind of nice control, we also included a different strain that is called a pathovar syringae, which is also able to infect Nicotiana benthamiana, but it's known to not produce modified viosamin. It has very likely a putative glucnec as a terminal glycan residue. This strain is completely not recognized. Even if you incubate it with B-gall, the bacteria are not recognized. They are very pathogenic, and it does not make any difference for the bacteria whether the plant carries B-gall or not. That's completely consistent with the role of B-gall in immunity only when the bacteria carry modified viosamin. So in conclusion, for this part, B-gall1 triggers the release of the flagellin elicitor. It's very likely upstream of a hydrolytic pathway that results in the release of the peptide that is recognized on the cell surface. We've speculated that this release probably involves proteases, but I'm going to talk about that more in detail. We have seen that some strains avoid this by secreting a B-gall inhibitor, and other strains avoid this by making a B-gall insensitive glycan. So it's a very interesting glycan warfare between the plant and the pathogen to avoid the release of the flagellin elicitor. And that makes a lot of sense because the flagellin elicitor is recognized by most angiosperm plants. So it's a very important mechanism, recognition event to avoid. Now, I'm going to talk a little bit more about the inhibitor at the end, but I want to add a few slides on the role of the proteases. So we thought, as many others thought, that of course the flagellin monomer carries that 22 amino acid peptide at the end terminal half, and it needs to probably be released from the precursor so that it can bind the flagellin receptor FLS2. And that would then released, that would then trigger that immune response called PTI, pattern triggered immunity. And just to make clear, B-gall we think acts upstream of that. So B-gall acts probably in the monomerization of the polymer, of the flagellin polymer. Now, to make a long story short, yes, there is a protease that is required for the release of this peptide, and we call this SBT5.2. It's a subtilase. I'll explain this later. But the same enzyme also inactivates the flagellin peptide, and whether it's in the precursor or already as a peptide. So it actually inactivates the flagellin. Just to support this with experiments, if you incubate flagellin in apoplastic fluids, it's very quickly degraded. And also the immunogenicity is very quickly disappearing. So within a few minutes, we see the initial response that flagellin, if you incubate flagellin with leaf disc, you get an oxidative burst. But if you start incubating first in apoplastic fluid for some minutes, then that oxidative burst is absent because the flagellin immunogenicity is inactivated. Okay, so then we studied how that occurs, and what we did is we analyzed the peptidomic mass spec data. So what we did here is we precipitated the proteins in 80% acetone that precipitates the proteins but not the peptides. So we took then the supernatants and analyzed that by mass spectrometry. And by doing so, and then mapping that back on the protein, you can see that there's a couple of clusters that appear. What we think happens here is that flagellin protein is first cleaved at certain sites, and then there are exopeptidases that nibble at both ends to create these stacks of peptides. We miss a couple of peptides here, but that makes sense because these are containing the O-glycosylation sites, and it also indicates that in this case, at least, the flagellin has not been fully deglycosylated, otherwise we should have seen those peptides. But what is more important are these regions here. There are 7 different putative endoprotease cleavage sites. I just want to zoom in on the peptide that's most relevant, the FLG22, which you see here in red. It indicates by the occurrence of these peptides that site number 1 would probably release it at the N-terminus, and maybe site 4 and 5 perhaps together would cleave it at the C-terminus. But I already mentioned that the flagellin peptide is very unstable. It's very quickly inactivated. So we also looked at possible cleavage events within the peptide. And you can actually see that there are several, and sites 2 and 3 are the most important ones. So what we then did is we ordered quenched peptides. These are octapeptides, or 8 amino acids, flanking each of those 5 different putative cleavage sites. These peptides are quenched, so they have a fluorophore, but also have a quencher. And they will only become fluorescent when they are cleaved. It's a really handy trick to zoom in on specific cleavage events. Now, if you incubate those peptides in apoplastic fluids, we can clearly see that they're all cleaved, but there is a preference. There's a preference for site number 2. Okay, so then we used this cleavage assay to go through the genetics to identify the relevant protease. So first, we transiently expressed EPI1. EPI1 is a subtilase inhibitor that is from Phytophthora infestans. And when we transiently express that and then isolate apoplastic fluids and then incubate that with quenched peptides, you can clearly see that all these cleavage events are strongly reduced in the presence of EPI1. So that indicates that subtilases are required for this. Now, the extracellular space or the apoplast of Nicotiana benzimiana contains a lot of different subtilases, but there are several that are more abundant than others. So we decided to silence the most abundant ones, the top 4 most abundant subtilases. And that worked quite well, because if you silenced them with virus-induced gene silencing and then isolate apoplastic fluids and then incubate those apoplastic fluids with these quenched peptides, you can clearly see that some of those, if you silence SBT5.2, you see basically the suppression of processing of all these different peptides. I would argue also quenched peptide 1 is one outlier here. The least of those is quenched peptide 3, and interestingly, that is even better cleaved when you silence SBT 1.9 and 1.7. And that makes sense because those 2 subtilases are so-called phytaspases. They cleave after aspartic acids, and there are 2 of those in that quenched peptide number 3 and not in the others. So quench peptide number 3 or site number 3 is cleaved by phytase bases, and the other peptides, other 4 cleavage events are caused by SPT 5.2. So then we took advantage of an SPT 5.2 triple knockout, because actually there are 3 genes that encode for this subtilase. And this was a line that was generated for a molecular farming project in my lab by Xinzhen Zhong by genome editing. And if you, when we isolated apoplastic fluids from these plants and then labeled that with an activity-based probe for serum hydrolases, you can see that there is a major signal disappearing. This is at 70 kilodalton. This is the size that you expect for subtilases. There are some weaker signals still remaining, so that must be the other subtilases are still there. And there are lots of other signals, and they are not caused by subtilases, but other serum hydrolases. So with this experiment, we demonstrate that SPT 5.2 is the main active subtilase in apoplastic fluids, and that's consistent with the proteomics data that we had already before that. Now, despite that, these plants look absolutely normal, so they don't grow in any different way, so they are, have no phenotype. But if you isolate apoplastic fluids from those plants and incubate it with these quench peptides, then indeed, very consistent with the silencing, we see that all the peptides are less cleaved, except maybe quench peptide number 3, but that's because that's more preferentially cleaved by subtilase, by phytase bases. Now, and importantly, if you then incubate those apoplastic fluids with the flagellin peptide, you can see that within 30 minutes, wild-type apoplastic fluids from wild-type plants will completely inactivate the peptide, yeah, but not when you take apoplastic fluids from the SPT 5.2 mutants. So that means that SPT 5.2 is degrading the peptides and inactivating it. So that means that in the absence of SPT 5.2, flagellin immunogenicity will increase. And to show that in a disease assay, we did a so-called priming assay. So we pre-treat the plants with a low concentration of the peptide and then infect with the pathogen. And we really need to go down to a level way lower than what is normally used, because the effect of the stabilization of a peptide will only be shown when you go to very low peptide concentrations. And indeed, we can see that at a low concentration of 10 nanomolars, we suddenly see that the SPT 5.2 mutants do not support bacterial growth as much as the wild type would, because in those mutants, that peptide is more stable and will induce an immune response. If you go lower than that, it becomes non-significant, and if you go too high to the classic 100 nanomolar, you basically overdose the plant with an immune response, so you don't see the difference anymore. Okay, so it is clear that SPT 5.2 inactivates the flagellin immunogenicity, and there are 2 different interpretations for that. One is maybe that is on purpose. Maybe SPT 5.2 dampens immunity to avoid the diffusion of the peptides to distant tissues where there is no infection, or to avoid the triggering of the immune response already after the time when the infection has been cleared. But I actually prefer a pathogen-centric interpretation, because bacteria may have adapted to infection and take advantage of host proteases to avoid their recognition by making sure that the elicitors that they produce actually carry a cleavage site for these proteases. And we actually have similar data on a similar peptide called CSP22, where we also noticed that this phenomenon occurs. So that actually indicated SPT 5.2 may not be really specifically involved in flagellin sensing or perception. It's more a general apoplastic protease that could, for example, be there to maintain protein homeostasis in the apoplast. And this is actually quite consistent with a study that I don't mention here, but I think it's relevant to mention. There was a study on the release of the flagellin elicitor in Arabidopsis that was also catalyzed by SPT 5.2. But also in their study, you can see that SPT 5.2 also inactivates this peptide. Okay, so I like to go back to this slide. So we have already discussed the proteases, but I really like to finish off with discussing the beta-galactosidase inhibitor. And I must really say this is a pretty exciting data set that kept us busy for the last 10 years. And I'm not exaggerating here that this has been the most exciting project in the lab for the last 10 years. This is a long project. Bala started this and T continued with this. You can see there's lots of collaborations that are on this work as well. And the beta-galactosidase inhibitor is called glycosyrin. And actually I visited T, started his own lab not so long ago, and I visited him in Bangkok, and in his hand you can see the inhibitor. Okay, so what is that inhibitor? So we do know, I just take you back to this slide to show that this is how we thought there might be an inhibitor. And we then characterize this inhibitor a bit further. We notice that it is a small molecule, it's heat stable, it's not volatile, it's polar, it's basic, and we know that it is secreted by the bacteria, but only when they grow on minimal medium. And that, of course, mimics the infection, so that's why that makes sense. So how do we identify this molecule? So we first wanted to identify which genes are required in the bacteria to make this inhibitor. And then we took I think a really a nice trick. We took Laxet, and you all know Laxet, right? Laxet is the gene that encodes beta-galactosidase, and everybody is using that for molecular cloning because if you add X-gal to the medium, you get blue colonies, right? So we thought, okay, take this gene and stick it into Pseudomonas syringae because then it will turn blue when it's growing on rich medium, and indeed it was white when it was grown on minimal medium. So that means that on minimal medium, the inhibitor is produced, and that inhibits the Laxet gene product. So then we did TN5 insertion mutagenesis and picked up mutants that were blue. So we picked up 300 mutants and sequenced the insertion sites for the TN5 transposon. And this is something that we did together with Gil Preston. So we identified 10 mutants that carry mutations or the transposons in a biosynthetic cluster called GSN for glycosyrin. So there are 5 genes here. The first is a dehydrogenase. The second is a RIP-D reductase, then a phosphatase, something with an unknown function, and a transporter. And when we mutagenized each of those 5 genes, we noticed that actually A, B, and C are essential for glycosyrin biosynthesis, and E partially contributes. Actually, C and E partially contribute. Okay, good. So then we, to confirm this, we already did what we call a Laxet inhibition assay. We took the Laxet protein and then incubated that with the supernatant of the bacteria. If the supernatant of the bacteria contain a Laxet inhibitor, then it would block the activity of this Laxet in an activity assay. So that's what you see here. You see that the mutant bacteria do not suppress Laxet activity, but the wild type do, because they produce the inhibitor. So this is a mutant that lacks the entire cluster. And when we put that on a plant, we notice that these bacteria are less virulent. That makes sense because they are suppressing immune responses, and that means that if you do not have this inhibitor, then you would not be able to suppress that immune response, and then you get less bacterial growth. So that makes sense. Okay, so if you take the mutant bacteria and put in the biosynthetic cluster back, then of course you get the inhibitor production also back. But interestingly, if you take now this same cluster into E. coli, then E. coli starts making the inhibitor. So that means that cluster is sufficient to make the inhibitor. Now, I just mentioned 10 mutants that we picked up, but there were a lot of other mutants. And when we analyzed which genes are disrupted in these mutants, we noticed that actually all these mutants map, or most of them map, into different regulatory components that are required for the transcriptional activation of this biosynthesis cluster. And that is because the biosynthesis cluster has a HARP box that is activated by HARP-L, and we found 12 mutants in this transcription factor. which is HARP-L is activated by HARP-RS, and we found 32 mutants in those two genes. And HARP-RS is negatively regulated by RHPR, which is negatively regulated by RHPS, and we found 50 mutants in that protein as well. So we resolved that regulatory pathway, but actually it was a lot helped by the fact that we already knew this regulatory pathway, because this is also how the type 3 secretion system is activated, and all the type 3 effectors, including AVR-PTOB. So it makes a lot of sense that this cluster is also switched on during infection, just like the type 3 secretion system and the type 3 effectors. And just to confirm that this is the case, we took a number of these regulatory mutants and looked at the expression of one of those biosynthetic cluster genes, and noticed that they are not activated when you grow these bacteria in minimal medium. They are not activated. This cluster, biosynthetic cluster, is not activated, and that's the same with AVR-PTOB. And if you take the promoter of this biosynthetic cluster and hook it in front of a luciferase, you can actually see that during infection, indeed, very early during infection, this biosynthetic cluster is switched on, and you see the activity of this cluster. Okay, so we have the biosynthetic cluster, and we then did a bit of bioinformatic analysis to see how frequently is this distributed. And when you just do look for homologs of the alcohol dehydrogenase, then there are quite a lot of microbes that have biosynthetic clusters that contain that ADH gene. And many of them are known to produce immunosugars. These are sugars that have a nitrogen instead of an oxygen in the ring. So we think our biosynthetic cluster is also able to make an immunosugar, but we also think it will do it probably a bit different, because what our cluster has is an GSNB, and that's a RIP-D homolog. And that's unique to a couple of those clusters for which we do not know what the molecules are, but they're not seen in any of the other clusters making the classic immunosugars. Okay, so we really wanted to know what that inhibitor was. So we went back to Laxet again. So now we hooked it up to a His purification tag, and we immobilized the protein on a nickel column. And then we saturated the nickel column with this Laxet column with metabolites that are produced by the wild type bacteria when grown on minimal medium. So we basically saturate the Laxet gene products with the inhibitor. We then washed out the non-binding metabolites, released it from the column, and then did cryo-EM in collaboration with Pinjun Zhang in Oxford. And they came up back to us with an amazing 1.9 angstrom resolution. So an atomic resolution of a small molecule bound to its target. And there it is. It's a small molecule. And we can zoom in a bit on that. This is a novel inhibitor because it has a 5-membered ring. Typically, other immunosugars, many immunosugars have 6-membered rings. But also we can see there are 2 hydroxyl groups most likely sticking upwards, and here there's a side chain that has that is branched, that sticks downwards. So we were guessing based on this structure that maybe the inhibitor that is produced looks like this. This is also combined with the information that we had on the biosynthetic pathway at this point. So that would mean a 5-membered ring with a double bond in it, and the nitrogen is there. And then this hydroxyl group would then presumably flip around and would occupy both electron densities. So a chemist started making this, Marcus Kaiser in Duisburg, Essen, a long-term collaborator. And he came back to me and said, well, we made it, but as soon as we put it in water, it is not stable. If we see a conversion, and we were puzzled in the beginning why that was. But then we realized that actually that double bond might be able to shift to this position, so you get an aldehyde. And aldehydes can be hydrated with a water molecule. And if that happens, then we get this structure. And that is exactly what we see in the structure. This is a hydrated molecule. And this is not something we expected because they are not supposed to be stable, but this is a stable dihydrate. And that's what we see in the structure. So Marcus made this molecule specifically, and we repeated the cryo-EM, and we found even 1.4 angstrom resolution structure. So it is really this molecule. We did inhibition assays. They are in the low nanomolar range. So these are very strong inhibitors for both LAC-ZET and bgal1. Now we can also now see why it is such a good inhibitor, because if you zoom in on how it binds to the active sites, you see that the nitrogen is close to the catalytic residue over here, yeah, and it perfectly mimics, and I find it really remarkable, perfectly mimics the galactose. So galactose is a 6-membered ring, but glycosyrin is a 5-membered ring, but it uses this dihydrate to mimic a 6-membered ring. So very, if you could talk about cleverness for small molecules, this is definitely a clever molecule. Okay, so we know what it is. How is it produced? Well, I already mentioned that quite unique to this cluster is the GSNB gene, which encodes a RIPD-like reductase. And RIPD is involved in riboflavin biosynthesis. So we looked a bit on the literature and started thinking maybe this is the precursor, because that is similar to the intermediate in the riboflavin biosynthesis, and it has 5 carbons and here a nitrogen. So maybe that is the intermediate that we need. This precursor is actually an intermediate in the purine biosynthesis pathway. It is produced by PERF and then converted by PERD into the next step. So we then made the mutants for PERF and PERD. Of course, they do not grow unless you provide them with purines to grow on. So we can do the assay. And we noticed that indeed the PERF mutant was no longer able to make the inhibitor, but the PERD mutant was able to make even more inhibitor than the wild type. And that makes sense because by shutting down PERD, you increase the flow of metabolites into the synthesis of the inhibitor. So if GSN, so this is the precursor, and that means that actually it's a novel iminosugar biosynthesis pathway because it branches off from the purine pathway, which is unique. Now, if GSNB then acts on this, then we expect that the product will be looking like this. Basically, it opens the ring, you get a hydroxyl group. We know that one of the other genes is GSNC, which is a phosphatase, so we speculated that that would then remove the phosphate. And then we need a GSNA, which is the dehydrogenase, that would convert this hydroxyl group in a ketone. Now, a ketone is very reactive. That will react with this amino group, and you get a ring closure, which is just normal chemical conversion. And then in water, we get the dihydrate that we see in the crystal structure. So this is the pathway, and we actually, I don't want to show all the evidence, but we were able to confirm this simply by making all the 4 enzymes. So the GSNA, B, and C, and PERF, together with a PRPP as a substrate, and all the cofactors, all in one tube. And then we had the inhibitor. And then we could actually show that every component was required and that they act using a trick with heat treatment. So we heat denature the proteins at different steps. We could confirm that this was the pathway and this is how the molecule is being made. Okay, so yeah, we know what it is. We know what this molecule does. So glycosyrin has this particular unique iminosugar structure. It's tiny and it's also multifunctional. So we know it's important in preventing the elicitor release by inhibiting bgal. And that's probably the main role during the infection assays that we do. But we did notice that when bacteria that are able to make glycosyrin compared to the mutants also induce the accumulation of other sugars, such as galactosylglycerol and trehalose. And we think that, and they accumulate quite high levels, so we think they may help the bacteria to hydrate the apoplast. We also noticed that the extracellular proteome is differently glycosylated, because by inhibiting beta-galactosidase and other glycosidases, you may change the glycans on the cell surface of the plant. And indeed, that's what we detect here. And it does so not by only inhibiting beta-galactosidase itself, but also by other beta-galactosidases, but also by even inhibiting alpha and beta glucosidases. And that is because it is a bit promiscuous, so it can also inhibit also glycosidases, other types of glycosidases. So in the years to come, I would predict that this molecule, there will be tons of other functions described to this molecule. It's not only that it will suppress glycosyrin, suppress flagellin recognition, but also other roles in the future. And it's actually, if you think about it, it's also quite a clever molecule because it's very small. It's very difficult for the plant to avoid the inhibition because it mimics the substrate almost perfectly. So it's very difficult to avoid. It's a very clever small molecule. And it's actually quite common. This gene cluster has been detected in many different Pseudomonas syringae strains, and we noticed that it is flanked by transposons and integrated at the tRNA insertion site. So we think it's moving around through horizontal gene transfer. And the strains that from which these glycosyrin producing strains were isolated, and the hosts are very, very different. And you can easily predict that for all these different pathogens, they will all make similar glycosidases. They will all be sensitive to the same beta-gal inhibitor, glycosyrin. So the impact of this small molecule in disease is much bigger. Okay, so I have now addressed these two questions. So we know why beta-gal is inhibited and how it is inhibited. But that's only the tip of the iceberg, right? Because there are many other hydrolases. So could there be other hydrolases that are suppressed? Just to mention one data set from Daniela. So Daniela has been a postdoc in my lab, now works in Trondheim, her own lab, and she mixed 3 different biotelated probes that target different hydrolases. And when she monitored labeling by purifying the biotelated proteins and then do mass spec, she was able to monitor 171 different hydrolases simultaneously in an aplastic fluid. And when she did that with comparing infected versus non-infected plants, she noticed that a massive change in hydrolase activity. So there are lots of hydrolases are activated upon infection, mostly are serum hydrolases. But we also see quite a few that are downregulated in activity. We think they are inhibitors, and that includes beta-gal1. And there are many other hydrolases that we are now picking up on, because you can predict that they are inhibited, and then the same questions that we had: how does it happen and why does it happen? So final conclusions is that DC1000 suppresses dozens of hydrolytic enzymes, including beta-gal1. It does inhibit beta-gal1 because that promotes the release of the flagellin elicitor, and DC1000 is trying to avoid this. SP5.2 is releasing FLG22 from its precursor, but also inactivating it, and that's actually quite an important role. And we think that is because bacteria took advantage of these of this protease to avoid recognition. And we identified glycosyrin as a novel iminosugar that manipulates host biology, glycobiology. Okay, I'd like to thank at the end my lab. This is when we are not dressed up in lab coats. This is Oxford. I'd like to thank the funders, the collaborations, and you for staying here. Thank you.
Thank you, Renier, for a very impressive talk. I'm impressed how you found out about the synthesis of the inhibitor. I don't know if there's any questions for Renier.
Thanks. Thanks for the talk.
I can hear you, but not through the mic.
Yeah, maybe.
Hola, hola.
This is magic.
Okay, I can make the question. So, regarding the, I remember that you showed some data that you can see the bacteria inoculation on the lip.
Yeah.
Yeah. That's a very nice experiment. So I would like to know what is the inducer in the bacteria for inducing the glycosyrin? Is it constitutive or is it just induced by something that you don't know?
I think it is known to some extent.
Right.
It's low pH and low nitrogen.
Right. I see. Okay.
Yeah.
So because I was wondering, what about if you just put the bacteria with the apoplast fluid? Is there something in there that stimulate the synthesis of the glycosyrins? That could be nice to do.
Low pH, low nitrogen.
Right.
Yeah. Yeah. Basically, they starve. That is why they trigger this.
Hi, thank you very much for your nice talk. I'm wondering whether you have analyzed also the apoplastic components of roots.
We didn't, but it has been done. Not on benzimiana, but on tomato. Yeah, it's similar composition. It is some differences, but globally very similar composition.
But this glycosidase is also present in roots in the same.
Yes, I'm quite sure it is. Yeah.
Yeah, and so do you think that that enzyme also has something to do with pathogens that attack the roots directly?
Yeah, you would. Yes, I think that would. Yeah, that might mean that the same thing will happen in roots. Yes, good question.
A second question, I don't know, maybe I miss it, but what happened with the flagellin when it's glycosylated? Is this sensitive, similarly sensitive to degradation by the peptidases?
Yeah, can't really distinguish that at this stage, but it is clear that the polymer is stable. The monomers are not. So the monomerisation is a way to release the ester. But the role of so definitely the glycans are important for that. But I don't think the glycans will protect against proteolysis when it is a monomer.
I see.
It will protect it when it is a polymer. Yeah.
Okay, thank you.
¿Alguna otra pregunta? No? I'd like to ask about the immunity because I understand that when you inhibit the so in let me think it properly. The bacteria is recognized. So the bacteria is producing the inhibitor to the to the glycosidase.
Yeah.
And by that, then because I remember you were telling us about the plant-centric view and the bacterial-centric view, because the immunity is being activated and that's convenient for the. No, it's been really. I'm a bit confused now, but can you explain a bit more about that? Because that's a bit counterintuitive,
Yeah,
right?
it is, but I think the way to see this is in evolutionary times, pathogens are in the advantage. They have gone through more cycles of evolution, so they may have adapted much better to the host than the host has avoided infection. So, and the way they adapted, because I think the same enzyme in Arabidopsis in the apiplast does exactly the same thing. So we see something very similar between Nicotiana benthamiana and Arabidopsis, which are quite different plants. So my prediction is that very likely those enzymes, they're not there in order to act in immunity. They are there for another reason. And it's a very conserved reason. So I think this is why I like to speculate that this is a proteolytic machinery that all plants have, and all pathogens will have taken advantage of that by avoiding recognition.
Yeah, so they are not there by the immunity, but they are also and by chance they are acting as immune regulators or immune responders.
Yeah.
Okay. That's interesting. Yeah.
I'm just thinking whether those plants that can be resistant to this bacteria are affected in the production of this glycosidase, or there is another mechanism of tolerance.
You mean plants that are resistant?
Yeah, right.
Yeah, they typically recognize bacteria in other ways, in ways that bacteria haven't been able to suppress.
It doesn't have anything to do with this mechanism.
No.
Thank you.
A very naive question about fungi. Do you know if pathogenic fungi, also from the apoplastic part, uses something similar? It can be any horizontal transfer of this type of.
We have no. You mean the glycosyrin produced by?
Right.
We have not seen any evidence in genomes. It doesn't pop up as a hit, so I don't think fungi took it over. I do think still. No, I think the argument. I'm not so sure if they would like to benefit from beta-galactosidase inhibition the same way as because the glycans used by fungi are very different from those used by bacteria. So I think that is maybe a distinguishing thing, and that would maybe explain why in bacteria you see the evolution of those molecules targeting beta-galactosidase and maybe not. But we cannot really say until we have done experiments to show which enzymes are suppressed during infection with fungi. So maybe it exists, but I think maybe there could be differences.
Hello? Yeah. I have another question about the application of the inhibitor for alpha or beta-galactosidases, because it's quite broad. Now it has. So have you thought of any useful application for that molecule?
Now these glycosidase inhibitors are actually used as drugs to treat metabolic disorders, for example, diabetes as well. So yeah, there are a couple of possible applications. That they are promiscuous is actually quite common for immunosugars. They're not that specific for a certain subclass, certain class of glycosidases. There is some promiscuity amongst them. Yeah. Because
I guess now you said you can just reconstitute the pathway to synthesize it in a test tube. So that would be easy to produce, maybe, in case someone. Yeah, although I mean, growing bacteria without a medium, without a rich medium, is also not difficult, and we get quite some inhibitor from that process. Yeah. And you can purify it now?
Not really, unless you have a lot of Laxet protein.
Okay. Well, I don't know if there's any other question. If not, well, let me thank Professor Renier and.
Thank you.